Good morning, everyone. My name's Alex Brown. I'm AHDB's Beef Breeding Project Manager. It's my absolute pleasure to welcome you this morning to the penultimate session of our Dairy Research Conference, which is focusing on dairy breeding and genetics. We've got three excellent speakers this morning to discuss work around our dairy genetic evaluation services, from research through to delivery, along with the value provided to the industry by our genetic improvement tools. We've got three speakers this morning. First of all, we're going to have Marco Winters on screen now, who is our Head of Animal Genetics at AHDB. He will be followed by Mike Coffey, who leads the Animal Breeding and Genetics team at SRUC and also heads up Edinburgh Genetic Evaluation Services, more known colloquially as eGenes. And finally, we'll be joined by Tim Harper, who is a Senior Consultant at Promar International. We're going to hear from Marco, Mike and Tim concurrently before opening up to a final Q&A session, and we expect to wrap up at around 12:30. Audience microphones are going to be muted throughout the session, but we do encourage you to send in your questions as and when you think of them. You can do this by entering them into the questions panel, and that should be shown on the right-hand side of your screen. You can use that panel also to flag up if you're experiencing any technical issues. If you could include the name of the speaker that your question is for, we would be extremely grateful for that. We'll be recording the full session and we'll make this available to you after the event so that you can revisit it at your leisure. At the end of the session, you'll receive a prompt to a short survey to provide us with your feedback, and we'd really appreciate it if you could complete this for us to help us continue to improve the sessions that we run going forward. So that's more than enough from me for now. So without further ado, I'd like to hand over to Marco Winters.
Thank you, Alex, for the introduction, and good morning, everybody. So I'm going to kick off this session on animal breeding and genetics, and it's really an opportunity for me to showcase, I guess, the improvements that we've made over the last year. And well, to remind you, if you haven't seen it yet, in the press or on your own herd genetic reporter and the bull catalogs, but also just to stimulate some thinking on where we're heading. And certainly in the later session that Mike will talk about some of the new stuff, and Tim reiterating, I guess, some of the messages again. Hopefully you'll pick some good stuff out of it. So the title that was given to me was to introduce a new index as Healthy Cow, EnviroCow and Gestation Length, but I'm going to throw in a bonus trait feed advantage, which was also introduced this year. So the presentation will cover all four and a little bit of extra background. So to start us off then, just to remind you what AHDB's involvement is in this area. So AHDB Dairy and Genetic Genomic Evaluation Services is responsible for offering that service to the industry. So when you look at your bull catalogs or when you look at the Breed Society web pages, etc., the data you're seeing on bulls, the production proofs, the health and fitness and type proofs are calculated by us and paid for by dairy farmers. Now that final point of being paid for by dairy farmers is important, because what that does mean is that we do provide independent data. We're not focused on any particular breed or any breeding company or for that matter even any country that's supplying genetics. For us, it's just highlighting which genetics are important and which are really the important genetics that dairy farmers in the UK should be using. But we don't do that on our own, and that's really the purpose of this slide is really to highlight that the reason we're successful is because we collaborate with so many international but also national industry partners, and that's really what makes it a great success in collecting the data on farm that are being recorded by farmers, collected by breed societies, milk recording companies, and shared with us for genetic evaluation purposes. So in the middle there you see our service partners, and Mike Coffey chairs or heads up eGenes, which sits in the middle there. I've also listed Interbull and ICAR, so internationally we very much are also involved in the international scene. And more as you go towards the right, the genomic partners are becoming increasingly more important to make sure that also farmers are genotyping through the various services that are being provided in the UK, receive UK evaluation that AHDB run through our genomic evaluation system. The next slide then gives you just one snapshot of how dairy genetics really has got an impact in the industry. This is looking at the genetic trends based on insemination data. So every year through milk recording, we gather about 1 million insemination records on the dairy industry, and from that we can track how genetics is progressing. And so here I'm illustrating the milk line, which is the blue line tracking up, and the other one is the fertility line, and I'm using here calving interval as a proxy for fertility measurement, and I know it's only a proxy, but it illustrates the point. So you can see that up until 2005, as milk was progressing very nicely through genetics, we were also seeing a deteriorating calving interval trend because these traits are genetically correlated in a negative way. So as milk production was going up, fertility got worse. But then in 2005, we introduced a fertility index to the industry, and we can see that pretty much immediately the breeding decisions changed, that farmers and industry were still selecting for higher milk genetics, but at the same time we could now select genetics that also improves our fertility, up to the point that the bulls we're using today are equivalent or actually slightly better in terms of fertility than the bulls we used in the 90s. So it shows how genetics can have a massive impact, and that's also all the more reasons why we really need to think about which genetic traits we introduce to industry to make improvements. So the next slide gives you an overview of all of the traits that we evaluate, and again, today we're not going to go into the detail, but it does show that as more data is being recorded on farms, we can really do an awful lot more, and that data really has significant and permanent improvements that we can contribute to the industry. And it's only by industry using the data we see that. And well, I've shown you calving interval as an example, and we do know that fertility on the dairy herd has improved significantly since we started using better genetics, and certainly we can see the same for other traits that we're tracking. The new traits that I'll be introducing now are listed on there, but I'll go into a bit more detail now on the 4 new traits. So the 4 new traits that we highlighted, sorry, before I go into that, I should actually just highlight the well, what we do in a pre-step, and Tim Harper will talk about that if you go to the next slide. Yeah, so the traits that we've got, well, the challenge with it is because there's so much data available, how do we really identify which are the best genetics to use on farm? And for that reason, we've constructed 3 different economic indexes, depending on the system that you're operating in. So we've got a PLI, probably the most well-known. It's an all-year-round calving system. It is a within breed ranking, but within that you can really highlight which bulls are going to be the ones that are going to make me the most amount of money over time, and actually, as we'll see later on, also help us to improve health and welfare and efficiency through carbon. The pie chart on the right-hand side there gives you a crude breakdown of the traits we include. So about 1/3 of it is driven by production, but 2/3 of it is really looking after the, well, how can we reduce costs on farm and not just focusing on improving output. But as well as PLI, we've got also the SCI, specifically set up for spring block calving systems. That is in the crossbreed ranking, so you can directly compare different bulls from different breeds. And then we've got ACI, similar to SCI in the crossbreed, but specifically geared towards autumn calving. And all of these are slightly different because we do know that those different systems do require different genetics to work. Okay, next slide, please. So now I'll then start talking about these new traits. In April, we introduced a Healthy Cow Index, and it's a financial index which will help us to improve the profitability through better health. We also introduced gestation length, probably more of a management tool to help us reduce gestation length if we want to narrow the block or get some cows to calve earlier. Then in the last proof run in August, we introduced Feed Advantage, which is a first for introducing a feed efficiency evaluation for the Holstein breed based on genomic data. And then EnviroCow, probably, well, very topical at the moment. We know that the industry is under scrutiny, and we're being challenged to reduce our carbon footprint. And we really felt that we needed a genetic index to help us also achieve that aim. So I'll talk about the detail in the next couple of slides. So the first one that I just want to highlight then is gestation length. As I said, it's probably more of a management tool than a breeding tool, but it's based on 7.5 million calving records collected in the UK. It's very heritable, 43%, so we do know that we can really make significant impact in reducing the calving into the gestation length if we're interested in it. The various graphs there are just indicating that when we look at first of all, the distribution between the different surface sires, we do see that some surface sizes give us a shorter gestation length, like 272 days on that chart. But also on the other side of things, we've also got bulls that give a gestation length of about 290 days. And then obviously that's a significant difference between the very, very extreme bulls. If you then look at the top right chart, what we also know is that over time, gestation length is actually decreasing. This data is mainly based on the Holstein breed, but we see similar trends in other breeds. And actually in the very recent years, of 2019 calvings, the average was about 280 days gestation length, and that's so it's a decline from about 281, 282 a few years back. We also obviously know that there's difference between different lactations and between males and females, and all of those things we got to correct for when we come up with the PTAs and predicted transmitting ability genetic indexes. So the next slide just well summarizes what we publish. So the PTA, the predicted transmitting ability that we publish, is presented on a day scale. So each day represents a day change in gestation length. And because we want to reduce gestation length, we therefore got to look out for the negative bulls, because those are the ones that reduce the gestation period and can obviously make an impact on gestation length on farm. And as I said earlier, and a few times now, we see it more as a management tool if you really are interested in calving a cow earlier, maybe to make or tighten up the block. Then the next slides will talk a bit more about healthy cow index. Again, an index we introduced in April. So it's similar to PLI, SCI, and ACI. It's made up of various traits that contribute to an improved health and fertility of our dairy cows. It's a financial index. There's a pound value attached to it, because we do know that if we improve the health, longevity, and fertility of our herds, it also contributes to an overall profitable herd. So the breakdown, as you see there, is just for illustration, but it shows that there's various traits that contribute to it to make sure that we target the traits of importance that we measure in the UK. Then the chart again, which looks at the trend again based on insemination data, just shows that actually even though we didn't have healthy cow available on its own up until recently, through inclusion in PLI and through the industry targeting these health traits, we have seen an improving picture since the early 2000s already. But we do hope that making this index available just reinforces that message that it is something that as an industry we should be tackling, and luckily, as you can see from the chart, are tackling. What we then did is also look at bulls that are available in the UK and see, well, if we group our bulls by healthy cow, look at the top quartile and the bottom quartile, can we see a difference in performance of progeny when it comes to some of these key traits that we incorporate? So the next slide gives you the breakdown of that analysis, and the 4 traits here that we looked at were lameness incidence, calving interval as a fertility proxy, mastitis incidence, and then finally lifespan. So in all of the cases, the green bars on the left-hand side are our top quartile bulls. So those are the bulls that rank best for the healthy cow index versus the bottom ranking quartile on the right-hand side, the blue bars. So first of all, when you look at lameness incidence, you can clearly see that the better healthy cow bulls also produce daughters that have a lower incidence of lameness, and that significantly showed up in the first lactation, but also continued throughout the lifetime. Then moving on to calving interval, we can also see that the top healthy cow bulls had a shorter and a better fertility than the worst ranking bulls in healthy cow. Similarly, for mastitis incidence, again, the top ranking bulls would show a lower incidence of mastitis in the daughters all the way throughout the lifetime, especially when it comes to older cows, that genetics are really kicking in, and we can see that difference coming through quite clearly. And then the final one is probably a very important one and a very significant one, is the ultimate lifespan of the daughters of these bulls. Again, our top ranking healthy cow bulls would give us a much greater longevity than the worst ranking bulls. So it's an index that can really help us to drive health, longevity, and fertility of the national herd. And we would encourage you to look out for the index alongside the indexes available to you already. So then if we move on to the next trait that we introduced in August, please go on to the next slide. The trait that we introduced in August was a feed intake prediction. So feed advantage is based on a study that was led by Binji Lee up in SRUC Mike, who talks later, and Dr. Raphael Marodi also in Edinburgh. And so we looked at genomic predictions for feed intake based on data collected at SRUC and based on the genotypes that are available to us. Next slide. So feed advantage, what it is in a nutshell is, it's based on the dry matter intake that's been recorded at Lang Hill at SRUC for over 30 years. So there's over 750,000 intake records. So that does mean that every cow would be checked in terms of how much she eats, and the feed intake would be weighed, converted to a dry matter intake equivalent. And we've got data on 5,000 lactations, about 2,000 cows. So it means we're not just interested in feed intake and first lactation, but we're also interested in how does that cow continue to eat throughout its lifetime, and really get a good idea of the genetic influence that determines the dry matter intake of these animals. It's a fairly well heritable trait, 18%, so it does mean that we can quite nicely determine which genetics really make cows eat more or less. Now, dry matter intake in itself isn't the goal, because we do know that if we want to drive production, dry matter intake will increase, but what we are interested in is efficiency. So do the cows eat less than they produce, and therefore are more efficient, which is good for the environment, but is also good for the financial contribution to the farm. So feed advantage, what we then do is it's a combination of two. It's the feed efficiency that we can target throughout the lactation, but also lower maintenance in general, because we still want to just increase cow size, even though they may be efficient. We want to do a combination of, we want them to be efficient, but at the same time also on a daily basis have a lower maintenance feed cost. And so those two traits are combined into feed advantage to give us cows that can produce well, but at the same time maintain that level of production, because we don't want to penalize production just for the sake of it. So how much feed can we save each year while maintaining that same level of production? And this expressed as a kilogram dry matter intake. The difference between the best and worst bulls is about 500 kilograms of dry matter intake per lactation. So it's a significant cost on farm that through genetics we believe we can target through this index. The next slide, please. Okay, so then moving on to the next index, EnviroCow, which is incorporating feed advantage, because what EnviroCow is trying to do is again target output, but reducing the inputs on farm, and therefore have a better carbon footprint for the product that leaves the farm. We do know that feed intake has got a very strong correlation to methane, and methane being the one of the main sources on the dairy farm that we need to tackle. And that methane we can convert into CO2 equivalents to get our carbon index. What we are targeting through the index is methane intensity, so we're not necessarily targeting carbon output per se, again, because we do know that higher producing cows per cow will produce more carbon, but actually when we then convert that to how much does that mean for every kilogram of milk or every kilogram of product that leaves the farm, some of those cows can be more efficient. So we're targeting methane intensity, which is the gross output per kilogram of product that leaves the farm. And it's based on work that was originally done in New Zealand by a team there at Abacus Bio, and we modeled, and actually Alex, who chairs the session today, was heavily involved in that initial work to try and translate it onto UK scale so we could implement it. Next slide. Okay, so again, like similar slides, just it gives you a crude idea of the makeup of EnviroCow. So essentially what we try and tackle is three main components. We want to drive the production of our animals, and that's about 1/3 of the weight. At the same time, we want to improve the longevity of our animals, because if these cows stay on herd on the farm longer, we've got less wastage through rearing costs, but also culling of older animals. And then the final one is that feed advantage element. So we want to have cows that are productive, live long, and at the same time are very efficient in converting feed into product. And that's really the main drivers. There's a little bit of tweaking through fertility index, body condition score, and calf survival, and they contribute, but they wouldn't be the main contributors to the carbon footprint from the dairy herd. Because of the non-standardized way, I guess, of collecting carbon data in the UK, we decided not to publish it at a gram of CO2 equivalent that we could save per product, but at the moment we've standardized it so that the better ranking bulls will be the ones reducing our carbon footprint on farm. And the next slide just gives you a feel for, well, what we believe we can contribute when we do the calculation, look at the genetic trend that we currently already achieve. So we've looked at the trends that we achieve at the moment through the traditional breeding, and it probably isn't a surprise to you, but PLI and SCI and ACI and EnviroCow are very strongly correlated, because we've already been breeding for better production, better longevity, better fertility. What EnviroCow does is just put a little bit more emphasis on that reduced feed intake and improved efficiency. But given the current trend, we do estimate that genetics can contribute around about a 20% reduction in carbon by 2040. So it's a very significant contributor to making sure that as an industry, we can reduce the carbon footprint. And therefore, we do believe should be of interest and should be prioritized when it comes to bull selection. Okay, next slide. So in summary then, well, I've illustrated that there's already a lot of indexes being made available to industry by AHDB, and that is thanks to the data that's being recorded on farm, and Mike in his next talk will talk about the need for recording more data again, and just to make that case. The 4 indexes that we launched this year I've talked about, and what I've given you there in the table is the summary of the approximately 1,300 Holstein bulls that are currently being marketed in the UK, and gives you a feel for what's the average for gestation length, feed advantage, healthy cow and EnviroCow, but also where do the best and worst bulls sit. And final column on that table gives you, well, what's the level to be in the top 100 for either gestation length, feed advantage, healthy cow and EnviroCow, and those are some significant numbers where we can make significant improvements. So I guess the final point, just to finish off with, is just to well, make sure that you use the data that we make available to you, because we do know that genetics really underpins performance on the dairy herd. Obviously, check out the bulls you're using on farm, but also look at the cow data. And if you are milk recording, just a reminder that through AHDB we do offer the herd genetic report, so you can check your own feed advantage, healthy cow and EnviroCow, depending on the data that's available in your herd. So please do look out for that report and check your own credentials to see, well, where can you make improvements. And I think the final slide just gives you the URL or the web address where you can find that info, where we've got more fact sheets. We've also got some more tutorial videos to help you understand genetic indexes and how to use the data on farm. And hopefully, well, this just reinforced if you'd already heard about these indexes, or hopefully stimulated your thinking of the next breeding choices you're going to make on farm. So with that, Alex, I'll hand back to you again for introducing the next speaker. Thank you.
Thank you so much, Marco. It's really interesting stuff. We've got plenty of questions popping up already, but please do keep sending them through. I'll now hand over to Mike Coffey, and he's going to discuss using mid-infrared technology to predict pregnancy and TB.
Thank you, Alex, and good morning, everybody. I'm waiting for my slides to appear. Here we go. That's good. Right, okay. And I'm going to talk to you a little bit about predicting new phenotypes, or predicting phenotypes using new technologies with mid-infrared spectral data, but particularly in this instance using machine learning. And in the next slide, you'll see why we do this. Next slide. There we go. So, as Marco said, we've been working with AHDB as a strategic partner in evaluations for nearly 20 years or so now. And over the years, what's happened is essentially the research work that we do is predominantly focused on using existing phenotypes or new phenotypes to produce genetic evaluations. As the years have gone by, there's an increasing demand by industry and farmers in particular to select animals for a wider range of traits, and I think it's fair to say we probably in hindsight have done the easy things, and we now have a range of indexes that Marco's talked about. We have new indexes coming along all the time. Clearly we have to go back periodically and look at existing indexes just to make sure that they're still valid and fit for purpose. But more importantly, we're always looking over the brow of the hill to see what's coming down the road next, to make sure that we have genetic evaluations in place in time so that farmers can respond to changing circumstances. And I think it's fair to say the current circumstances are really focusing about resource use efficiency, the environment, and disease. And so we're looking at new ways of picking up particular phenotypes. We, excuse me, we do a lot of salvaging of phenotypes, and these particular phenotypes that I'm going to talk to you about today are phenotypes that we salvage from another process. And you'll see in the next slide what that process is. Sorry, you'll see it in the following slide. But in this slide, the value of TB is such that if we can produce better, more phenotypes that are better, more available more often, then we should be able to increase the impact of TB. But you can see with the existing index that we have here that the offspring of bulls that have good TB advantage figures are more resistant to TB. But the nature of the trait and the disease means that it's difficult sometimes to see that coming through in practice. So actually we've gone back one more generation, and we can see by looking at granddaughters of the best and the worst bulls from this slide, you can see that the better the TB advantage, the lower the number of daughters infected. But if you look at the box on the left-hand side there, you can see that the best 10 bulls had a TB advantage of plus 1.9 and had about 130,000 granddaughters between them. And there were about nearly 6% of the granddaughters infected. But in contrast, you look at the worst 10 bulls, about 10% or nearly 11% of the granddaughters were infected. So going back, if we could turn back time, and all of those farmers that used the worst bulls, because they didn't know at the time, we didn't have TB advantage index at the time, if they had used the best bulls rather than the worst bulls, there would have been around about 3,800 fewer cows infected. Now, clearly that means there's fewer cows infected, so there's actual fewer cows lost, but it also means that those fewer cows infected would infect fewer other cows. So it's more likely that there would have been less spread of TB within the herds had those bulls been used at the time, had TB advantage been available at the time. So you can see the impact of TB and TB advantage. And what we're looking to do is see if we can improve that impact by using this new technology. Next slide. So most of you will be familiar, if you milk record, with mid-infrared technology. And basically the milk sample that you take at each milk recording goes off to the lab, and it goes through an industrial photospectrometer. That's the machine in the middle there. And all it does is shine a light of a given wavelength through the milk sample, and whatever is reflected or absorbed, it's just one or the other, is dependent on the chemical bonds between the molecules in the milk. And clearly, in this case, it's been trained on fat and protein percent. And you can see that that's how you get your fat and protein percent. And the prediction equation there is 99% accurate. So it's a cheap way of predicting the fat and protein percent in the milk sample. Now, having gone through all that process, the interest for us is what other things could we predict? And you can see we predict fatty acids, energy balance, ketosis using that. Next slide. And obviously, we've taken that further on now, and as well as through routine milk recording for fat and protein, as a result of a project we ran with a retailer, we now can predict individual fatty acids. And the interest at that time was things like saturated and unsaturated fats. But we realized soon after, actually, it was more powerful for predicting other things. And in this case, it's energy balance, and that's available on the National Milk Records website for milk farmers milk recording. So it begs the question is can we detect other physiological or biochemical signatures from the milk spectra? And that's the whole premise of this technology, is that we have this sample already on cows that are identified. We know lots of other things about these cows, and the question is, can we predict other traits from them? because essentially those other new phenotypes would be free, and hence the phrase that we often use is a phenotype, because we already have the sample. The phenotype that we predict from it is essentially free. Next slide. So then we go on and say, right, okay, now if we can get milk spectral data from milk samples, and we currently receive from NMR around about 1 million per night, we have hundreds of millions. In fact, we have 33 billion spectral records from NMR. We receive them every night. And we make predictions from those spectral data about a number of different phenotypes. Now, some of the traits that we're interested in the future, and pregnancy and TB are examples of traits that you either are pregnant or not. You either have TB or you don't. They're called categorical traits, 0, 1, yes, no. And the existing statistical methods that we use for determining whether a fat percent is 3.8%, 3.9, 4, 4.14, these are continuous traits. And the existing way of analyzing those traits is not very good at doing it when the trait is a binary trait. And ironically, machine learning is very good at looking at patterns and discerning whether something is or is not. And so we started looking at it and saying, well, can we use it to predict other things from milk spectra? Now, machine learning is a kind of a wide phrase, is used all the time. It's also sometimes mixed up with artificial intelligence, and you'll know more about it in terms of autonomous cars and drones and Facebook, facial recognition, and all those kind of things. Deep learning is a subclass of machine learning, and it's the one that we use. And it's creating a lot of noise, as I mentioned. The question is, can we use it in agriculture? And is it better than what we currently do? And that's an issue for us because we have limited resources. And if we spend time looking at machine learning, we can't spend time looking at developing traits in a traditional way. So we have to be sure that it's going to deliver some kind of benefit to us. And we've used it to investigate a number of things like goat udders, horse obesity, and sheep CT scans, which I'll show you an example of in a little while. But the purpose of this talk is about fertility or pregnancy and TB status, which we'll go on to in the next slide. So those of you that milk record and that milk dairy cows will be very familiar with TB, and this is the process. This describes what we're trying to do. So the objective is to try to identify animals earlier than the normal circumstance. So if you start on the left-hand side, we see that a herd is under restriction as the breakdown starts, and it has to go through some skin tests. And if an animal fails the skin test, the reactors are removed, and then 60 days later you have to have another skin test, and that keeps going on for 60 days at a time until 2 successive skin tests are completely clear for the whole herd. Now, the test itself has a lower than is desired sensitivity, which means some cows that pass the skin test are actually infected, and so they remain in the herd in the 60-day periods between tests and can infect other cows. And what we're looking to do is to use the milk recording, the routine milk recording, to perhaps alert the fact that cows are affected or infected but have passed the skin test, and then the farmer can choose to either isolate them from the rest of the herd or get rid of them early, or a number of management decisions can be taken with that knowledge, that information. Next slide. So the results that we've had using machine learning, and we have access to all of the national TB data from APHA. And here's an example of the process using a few thousand animals. And this square on the left is called the matrix of confusion, ironically, and perhaps interesting. And what it's showing is the top left box is saying that the actual score is a 0, which is a pass the skin test. And down the bottom is the 0 that we predict using the machine learning. And you can see that in most cases, we're correct. But there are for those animals in the bottom left-hand corner, they have been they failed the skin test, and we have predicted them to pass. Okay, so that's a bit of a problem. The bottom right-hand corner is the ones where they've actually failed the skin test, and we've predicted them to fail. But interestingly, the ones that we're most interested in, the top right-hand corner, are those animals that have passed the skin test, but we have predicted them to have failed. And they are the ones, we call them the grey cows, and basically they're the animals that are floating around the herd that are infecting other animals, which you don't want, basically. And you can see from our initial results, which have led us to believe that we can do something with this, we get accuracies and sensitivities and specificities approaching that of the skin test. Next slide. Here we go. So then we go on and look at predicting pregnancy. And this was a result of working with National Milk Records, where they said they were interested in the possibility of detecting late gestation pregnancy loss. So this is a pictorial example of an animal that's cycling early in lactation. She's been inseminated around about 100 days, or confirmed service. And then the first yellow line is the confirmed pregnancy. So we know the cow is pregnant. And then, of course, later in lactation, she comes bulling again and has lost the calf, and then gets served and is pregnant again. And these animals are often a problem for the herd, because their calving is outside the routine cycle. Often, if they're very much later than this in lactation, sometimes it's not sensible to put the cow back in calf and she becomes a cull. But the customer, in this case NMR, was saying that sometimes these animals are unobserved by the farm for quite an extended period, and it's only when they come bulling again late in lactation that the farmer is aware that they're actually open and is then faced with a problem of what to do. And so what we're interested in there is, can milk spectra be used for the routine milk recording to pick up those cows? Next slide. And we find actually that using our research, we can actually get a validation accuracy of 88% or so. And when we apply that model to unseen records, that is at the top, hundreds of thousands of records have been used, but we don't use those records to validate. We look at unseen records, and the inference accuracy becomes 88%, which is similar to the validation accuracy, which is really good. But actually, when we collapse the different categories of pregnant to pregnant, non-pregnant to pregnant, and so on, we can actually raise that accuracy to 94%. And all of this is within the data that we already have access to. And it shows such promise that both of them now are subject to field trials. And in the case of TB, there's a field trial running down in the southwest and in Wales, where specific herds are being monitored using their milk records data to see whether or not in real time we can actually predict which cows have got TB. And aside from that, we're working with NMR on a field trial to see how many cows we can predict have lost their pregnancies using milk spectral data. So both of these are new phenotypes, and should they prove to be successful, the predictions would then find their way into evaluations. And so what you have then basically is far more records on TB in the TB evaluations. The accuracy of TB Advantage should increase, and therefore the impact of TB Advantage should increase, leading to acceleration of selection for resistance to TB. And clearly in the case of pregnancy, that gives farmers tools to make decisions. Next slide. So where do we go next with machine learning? Now, clearly it does hold a lot of promise, and we are extensively researching. We've done some work on extracting phenotypes from images. And this is an area of interest for us because we recognize, with all the developments in technology and so on, that farms will be populated with very many more cameras, essentially, but devices that take images of animals all day, every day, going around their business, And the question then is, can you extract data from those images that can be used in breeding? We've shown that we can do it in goat udders, for example, with about a 96 or 7% accuracy predicting goat udder scores. So the obvious question is, can we do that in cow udders, and therefore have more accurate selection for appropriately shaped udders? I'm going to show you in a minute about CT scans. We've written an app on horse obesity, so you can take pictures of horses and see how, and the machine will predict how fat they are. There are devices called near-infrared and hyperspectral cameras that can be used to take images of things like meat quality and other characteristics, and they have an area of interest for us. But clearly one of the other areas are other traits, and those other traits would be things like Johne's disease, BVD, improvement in things like mastitis. But of course, the other thing is that we wonder whether or not the signals that we're detecting are strong enough to be able to be picked up from the bulk tank. So rather than do individual testing, the bulk tank is tested every single day. And so we could analyze the bulk tank sample and reveal some underlying issues that may need to be investigated further. Next slide. And this is an example I mentioned of CT scanning of sheep. We have a mobile CT scanner. On the left is the actual image taken by the CT scanner, and you can see this is a male. The image in the middle is the one that's been processed by a human, where they manually, using a computer software, remove the cradle that the animal is sitting in and any extraneous superfluous appendages that don't contribute to the picture. And that's the middle image. And you can see on the right is the image that's being created by the machine learning. And the image on the right and the image in the middle are almost indistinguishable, even at the pixel level. So machine learning is really good at extracting images from, in this case, CT scans. Next slide. So the conclusions on machine learning is that it's a new technique with lots of promise, and it can create new phenotypes for genetic evaluation. But you have to be aware of false prophets here. It's very sensitive to the accuracy of phenotype, so we still need recording. And for those traits that are of interest, such as methane emissions or feed intake, they will have to be recorded as well. It's very good at using sensor data, so all the new gadgets and technologies that are going to be coming on will provide a wide range of applications in agriculture. And it's often used in edge computing, and this is where you have a device in the parlor, for example, where data is recorded locally and then a prediction is made in real time. And so the animal, the gate at the end would open and the animal would be shedded out into a holding pen where it could be reviewed and something happened to it. So it offers a very great deal of promise in terms of predicting new phenotypes and improving genetic evaluations. And I think, next slide, I think that is it from me. If you want to know more, there's some information. They're the papers that we've already published. Thank you.
Thank you so much, Mike. It's some really fascinating work there. Do keep sending your questions in for Mike as we continue. But for now, I'd like to welcome our final speaker, Tim Harper, who will be talking to us about what the economic returns are from improving genetics. Thanks, Tim.
Thank you. Good morning, everyone. Yeah, I'd just like to talk about a project that we've been involved in 3 times since PLI came around, working with Marco. So we're really looking at the economic returns from genetics using PLI as the primary measure. So we first did this project back in 2008 and repeated it in 2011, and we've just updated it using the current PLI. Obviously, PLI has evolved during that period, so we've just updated it using the latest figures. 2 primary purposes of the work that we were doing. Firstly was to generate statistical analysis between genetic data using PLI, but also using a lot of the other genetic metrics, which Marco was referred to in his presentation earlier, and really looking at marrying that up with real world technical and financial data on a whole herd stroke farm basis. And I think that was very much with a view to helping Marco and the industry move forward in terms of the genetics and the indices that we're actually using. The second thing we wanted to do was actually to justify in more straightforward terms, just looking at a straight economic relationship between PLI, which we were using as the key composite genetic index, and really to encourage its greater adoption by dairy farmers. I think if we go back to the time when we did the first study, PLI had come into place in around 1999, and that was sort of 5 or 6 years in, and we were getting to a point where we're trying to encourage its uptake further as the primary generator, and part of this was to provide some insight into how we might encourage it to be adopted more widely. Thank you. Next slide, please. So the methodology we've used for this is fundamentally fairly straightforward. We've used financial data from our Promar Farm Business Account Service. In the latest case, we've used data for the financial year 2019-20, and very straightforwardly, we married that up with PLI and the other genetic indices and component parts using the data from the milk recording services. The latest study, we used 410 Holstein herds representing over 104,000 cows. It's fair to say we focused on Holsteins and we focused on PLI just because the weight of numbers that were available to do that analysis was so much greater than other breeds or using some of the other genetic metrics. So we focus very much on that overall picture in terms of trying to get a valid relationship. So the first thing we had to do, and we've kept to the same methodology as to the two previous studies, and this takes us back to when we did the first study, was to devise a methodology by which we would measure the genetic basis by which improvements in profitability were made. So to do that, what we tried to do, we sat down with Marco and colleagues at AHDB, and basically we tried to look at the aspects of income and expenditure on farm which genetics are most likely to affect or to impact. Thank you very much. So these were the metrics that we decided upon in the end. Clearly there was quite a bit of debate around some of these, whether they should be included or whether some of the ones that weren't should have been, but we felt these were the ones that are recorded as discrete items where genetics are most likely to influence. So milk income, obviously that's a combination both of output and also of milk price stroke component parts and seasonality, although the vast majority of these herds are all year calving, so seasonality would have played quite a limited effect on the overall picture. Calf income and cull cow income. Cull cow income would also include income from TB, compensation cows, etc. So any income generated from cows leaving the herd. Obviously a cow leaving the herd dead would be a zero in that case. And that cull cow would be part of what we'd refer to as the herd replacement costs. And then on the cost side, we looked at all the concentrate feeds coming onto the farm, but also any that are grown on the farm, which are predominantly going to be wheat and barley that farms are growing on farm and feeding as such. Also looking at purchased roughages, which the predominant feeds in that category tend to be brewers' grains and Trafford, Gold, or the various similar feeds to that, and also any purchased silages and hays where the farmer is looking to supplement that that's been grown on the farm. We also included direct forage costs, which included seed, spray and fertilizer. We looked at the cost of replacement animals, which basically are any animals joining the herd, whether they be purchased or whether they be reared on the farm. And they also form part of that herd replacement cost. We decided to include vet and medicine costs, and obviously AA and semen, because that would be one of the most direct costs of all in terms of trying to measure something that genetics are going to influence. Thank you. Next slide. So I've just put a repeat on the right-hand side of this slide of the figures that Marco showed earlier. Obviously, when we did these previous studies, PLI and the component parts were different. I think in the earlier days, the production part would have still represented a greater proportion of the whole, whereas that's now fallen back to 34.4%. And then we've now got all these other components which have mostly grown in the period over which we've been doing the studies. So basically, the project outputs we were able to deliver, firstly, was a statistical analysis for Marco, whereby we actually looked at the correlation of the financials using all of those individual metrics which I've just run through, but also the composite figure in terms of the genetically influenced margin, to correlate those with all the composite traits, particularly within PLI, but also to look at a lot of the component parts within it as well, and also to explore some of the relationships with some of the new genetic metrics which have been put out and which Marco was talking about earlier. Secondly, we looked at a simple summary, which I'll go on to talk through in a second, looking at the overall economic analysis based on PLI, and that's been disseminated recently both through AHDB, and if you've been reading the farming press, there's been quite a few articles picking up on it as well. And yeah, just a reminder there that the PLI has been updated twice since the prior studies, so it's quite difficult looking at how the changes with profitability have been affected because PLI has moved in the meantime. Thanks very much. Next slide. So in summary, this is the part where we're really just looking at a very simple takeaway message in terms of how much profitability measured by genetically influenced margin per cow is generated by each point of PLI, and using the latest analysis using the figures I described, it works out at £1.58 per point of PLI. Marco's done some projections on that for a typical 150 cow herd using the top 10% UK herd figure with an average PLI of 197 and comparing that to the same sized herd of average genetic merits, currently around £70 PLI. And if you do that, the top herds are realising a genetically influenced margin of the £1.58 per point of PLI. And then secondly, I've grouped all the herds here, looking at top 25% herds, looking at the average and looking at the bottom 25% in terms of their PLI. So they're the results which I'll explain on the next couple of slides. Thank you. So this is really focusing on the big picture income items and also a bit about the types of herd involved. So you can see within the averages, we're looking basically at high output herds within this analysis. And as I say, the vast majority of them are all year calving. You can see one of the slight variations is in terms of milk price, 30.47 for those with the better genetics. 30.39 and 30.12, and this was one of the factors that's limited our ability to do this work. So obviously milk price can be significantly influenced by a farmer's milk buyer. So what we've tried to do with these studies is to carry out these studies when milk prices between milk buyers have been relatively level, so that we weren't getting a lot of bias in terms of external factors which weren't to do with genetics, but which inherently we had to include within the analysis. You also see that the farms with the better genetics measured by PLI tend to have the larger herds. So the average herd size for the top 25 was 303 cows compared to the average of 254, and the ones with a lower genetics measured by PLI had 196 cows. So if we look at the income items there, the milk income is 3255 for the top PLI herds against 2972 for the average and 2576 for the bottom ones. So it's interesting that although production's now only accounting for 34.4% of the PLI within the overall metric, we're still getting a lot of the significant difference in terms of performance is actually being shown through greater milk income, predominantly through milk yield. The milk price will be a combination of some variation between milk buyers, but obviously it will also be affected by components and also by seasonality. Interestingly, the calf income, the herds with the better genetics measured by PLI, they're generating calf income of £173 per cow. Compared to 155 on average and 146 at the bottom. This is the average per cow, not per calf. So obviously a cow that's calving more frequently and has better fertility, she will be potentially generating more calves per annum than a cow that's calving or a herd that's calving less frequently. So the calf income, better fertility will also be positively influencing that figure. And so total income with the herds with the better PLI measured by top 25% had 3428 compared to 3127 average and 2722 for the ones with the lower PLI. Next slide, please, where we'll look at the costs associated with this. So the second line down there is the income carried forward. And these are the different costs associated with the herds in terms of the same groupings. So the top line there is all the concentrate feed and also the purchased roughage. So they are spending significantly more than the average, and again compared to the bottom. Direct forage costs actually very, very little across the groups here. So you can see, if anything, the ones with average PLI are slightly higher than the farms at either end of the scale. Herd replacement costs, they are slightly higher, 273 for the farms with the better genetics. This is going to be a combination of all the factors I mentioned earlier in terms both of cow income from sales, but also the cost of rearing replacements. But only that is, only £3 greater, as you can see, than the average herds, although the bottom herds by genetic merit, £30 less. Vet costs, again, slightly higher here for the top 25% herds than average, 76 at the bottom. And here's where we can start seeing this is something that's probably not going to be entirely influenced by genetics. A lot of the veterinary costs, and an increasing proportion of them we see now, are more for advisory work and for effectively disease prevention, and hence within that category, this isn't just a farm spending more on drugs if they've got better PLI. A lot of that will be the same types of herd who are doing preventative measures to actually avoid clinical disease. And finally, AI and semen costs. Here we've got £54 for the top 25% by genetics. So they are investing more heavily in AI and semen, although the AI costs will be influenced in terms of methodology as well as just the semen cost itself. So overall, the genetically influenced margin per cow for the farms with the better genetics came out at 1,818 compared to the average herd of 1,670 and the bottom of 1,529. Thank you. Next slide, please. So I think it's important, just in drawing this particular presentation to a conclusion, just to say there are 101 factors that will influence profitability. So we have to be very conscious when we're looking at output such as this, that genetics will only be one influence, but it's probably one of the, and I think in consultancy business, one of the things we're looking to do is to identify factors that are most limiting performance. And I think on a lot of farms, genetics probably in the past, hopefully less now, will have been a key limitation in terms of farmers achieving the level of performance they want to do. So I think it's worth looking at the figures in that context. And if we're looking at what is likely to be the one of the other key reasons is the capability of those who manage and work on the farm and who enable the cows to achieve as close as possible to their genetic potential as possible. And we did do a piece of work using a very similar data set for this for a colleague's PhD. And what he was looking at was essentially not so much the genetics of farmers, but he was looking at their behaviors and their attitudes. And by looking at the very top performing farms, looking at how farmers actually influence performance, and some of those findings came through very strongly. So if we can marry up very good genetics measured by something like PLI, together with farmers who have good attitudes and behaviors with regards to actually achieving profitable farms, they're going to be the guys who are right at the top of the list in terms of overall performance measured by profitability. It's probably also worth drawing the session to a close in terms of looking at how the figures that we've looked at by genetically influenced margin translate to actual paper profit. And I think what the line that we took was, if the other factors aren't influenced by genetics, most of those benefits that we've seen in terms of genetically influenced margin will translate directly through to the bottom line. And I think I can be very confident that by focusing on those key criteria within this metric, that will actually be the case. So I think in terms of final conclusions, I think we're very confident, and I think this stemmed through from the two earlier studies. I think we can very much provide an endorsed reassurance that by using PLI as a primary selection index, dairy farmers can be confident that they can use genetics to help drive their profitability. I think that was a measure that came through strongly, and hopefully through a lot of the messaging that we did at the earlier projects, that helped endorse PLI as a metric by which dairy farmers could actually make selection criteria, and they can still choose using other traits if they wish, but by actually using PLI as a filter for those who are all year calving as their primary filter, they can then actually be confident in terms of the fact that overall they will achieve greater profitability through the use of genetics. So thank you for your time.
Thank you so much, Tim. It's really pleasing to see that the work that AHDB Dairy do with SRUC in developing these genetic improvement tools do translate directly to value for the industry and for the levy payer. I'm going to open up for questions now. So thank you so much to everyone who sent their questions through during the talks. Please keep them coming. We'll cover as many as possible in the time that we've got left. My colleague Brian Nicholson has been monitoring the question panel throughout the session. So at this point, I will hand over to Brian. Thank you very much.
Thanks, Alex. Good afternoon, everybody. We've got quite a few questions. Thank you very much for them. I'm going to go straight into the question session and to Marco. Marco, first one, how do I compare Irish bulls to British breeds when they are marketed with different indices? And why does the island use different indexes?
Okay. Very good question, and it is one that we get quite a lot to say, well, how can we compare different bulls from different countries and make sure that we select the right genetics for the UK environment? And there's actually a very simple answer. So every bull that is marketed in the UK has got a UK genetic index. So if the breeding company you're dealing with only provides you with Irish figures or US figures or Dutch figures for the matter, then please do get in touch with them again and challenge them why they're not publishing the UK data, because all of these bulls are through our international partners at Interbull. We share the data with all the countries. So Ireland would send the data to Interbull, we send the data to Interbull, and Interbull then provides us with a international rankings to say, well, how do these bulls rank on the UK system? So every bull has got a UK index, and certainly if you're interested in using Irish genetics for spring calving systems, I would encourage you to ask the AI company for the SCI figures, the spring calving index figures, or if they're not willing to provide you that, you may want to look elsewhere, because you may want to question why they don't want to share that data. But that data is also available on our website to look at it. Now, the reason there is differences between different countries, because we did actually do a study with New Zealand, Ireland, and the UK where we compared our three indexes, and we know that each of us will rank bulls slightly differently. And that's not because we're necessarily thinking these bulls are performing differently, but the main reason, and that actually goes back to the work that Tim also highlighted, is that UK economic conditions are different to Irish and New Zealand. Now, we all grow grass, but also the way the milk payment schemes work are slightly different. Concentrate prices may be different. Calf prices may be different in terms of what they can generate from income from calves or cull cows. And all of these things we've got to consider. The same is true for somatic cell count penalties. They differ between countries. And so SCI is very much focused on UK conditions, UK economic circumstances, which type of genetics work on the UK system. By all means, look at other country indexes, but do benchmark against how bulls would rank in the UK, because that ultimately is what we're trying to improve.
Thank you. Next question, is it possible to buy a group of bulls that could shorten calving block by 1 week, and this could be used across the whole herd?
It's possible, yeah. Well, we know that the difference between the best and the worst is approaching 10 days. So it depends obviously a little bit on the type of bulls you're already using currently. If you're already using average bulls, you can probably shorten it by 4 or 5 days in your block. But yeah, it's very much possible to look at that index and select bulls that will improve it.
Okay, thank you. Next one, can EnviroCow apply to crossbreeds or lower creatine breeds used in low input systems? Yield could possibly balance health traits and optimize farms and select high yielding Holsteins.
Yeah. So whereas PLI is within breed, SCI and ACI are crossbreed. When it comes to EnviroCow, currently we've produced as a within breed index. So within each breed, you can identify the better bulls versus the bottom ranking bulls. We haven't produced as yet a crossbreed EnviroCow, and that was mainly because we were conscious that we didn't want to confuse the message by coming up with a PLI EnviroCow and SCI EnviroCow. But at the same time, if the interest is there from industry, and we believe that the interest is there, it probably does mean that in time, and maybe next year or the year after, we will get this a crossbreed EnviroCow. But for the moment, only compare bulls within breed and identify the bulls within each of the breeds that are good if you're crossbreeding to help that improvement. And you're right, the different farming systems will have slightly nuanced differences, similar to PLI, SCI, ACI have slightly differences. But in general, we'd be fairly confident that the bulls who rank highly on EnviroCow would work, would do the best job in all systems.
Okay, thanks, Marco. For an EnviroCow, there's very little emphasis placed on fertility. How will longevity increase if the cow is infertile?
Yeah, no, it's a good question. And the reason for it is really because ultimately, if the fertility is poor, the longevity will be poor. And so the direct measure of when it comes to the carbon footprint is improving reducing the culling on farm and reducing the number of replacements on farm. And so the goal trait is really longevity. Obviously, if fertility is poor, if lameness is poor, if mastitis is poor, all of those will influence the lifespan of that animal. But the goal trait is lifespan, and that's what we incorporate in EnviroCow. When it comes to economic reasons, PLI will incorporate all of the disease traits individually because there is a financial cost to having mastitis or lameness, but it's not necessarily a direct environmental cost other than reduced productivity or reduced culling or increased culling and reduced longevity. So it's in there, but it's indirectly rather than a direct weighting incorporated.
And if the next question, if I do not, if a farmer doesn't milk record, is there a way he can work out what genetic weaknesses I have with the herd, have with the cows?
It's more difficult, but well, the easy option would be to genotype your animals. Even if you're not milk recording, the genotyping and getting a genomic evaluations would highlight the genetics and would give you, well, almost just as accurate a picture, maybe not as much on the milking cows, but certainly young stock would be fairly similar. You crudely can look just at the bulls you've been using in recent years. So take a look at the bulls you've used and of which you've got offspring milking in the herd. Look at their genetics, and that should give you an idea of the type of genetics you're milking. And
following on, how do the new indexes correlate with milk PLI?
EnviroCow has got a very strong correlation. Top of my head, it's greater than 0.9, so 90%. And if you look at the top PLI rankings, you'll see that the majority of them have got a very good EnviroCow index. Similarly, we tend to find the top PLI bulls have got a good feed advantage, but that's not always the case because feed advantage is a combination of feed efficiency and maintenance feed cost. And we do know that some of these higher producing cows are almost too big, and we do want to penalize them. So feed advantage will be lower correlated. So it's an important trait for farmers to really keep an eye on. Healthy cow, again, because health is already a strong part in PLI, that there's a positive correlation, and it's probably around about 70% correlation with PLI. Gestation length is pretty much uncorrelated. You'll find a mixture of bulls ranking in the top PLI rankings, or SCI or ACI for that matter. There is some discussion about whether gestation length should be incorporated into PLI, SCI or ACI. Potentially within the block calving systems, we could incorporate it because there's an obvious financial interesting in making sure that the block maintains that it is, but it's more of a management and a breeding tool. So we're debating that at the moment.
Okay, and then I might come back to you, Marco, but last one for now. How should you use the Healthy Cow and EnviroCow as part of the bull selection?
Yeah. So our advice is to use it alongside the existing indexes. So the primary driver should still be your overall profitability, so PLI, SCI, ACI. But once you've shortlisted your bulls, then look at their Healthy Cow and EnviroCow to identify the bulls that can do the best job for you. Now, if you're really suffering on farm with health and fertility and longevity, then keep a stronger focus on Healthy Cow. EnviroCow, it depends a little bit how the market is moving and where the signals are coming from. It's a little bit difficult for us to say, well, sacrifice your traditional traits because you want to improve the carbon footprint. I think the market will dictate. Now, the good news is that the correlation is strong, and I would encourage you to keep an eye on it, because the likelihood is that in a few years' time, and remember, matings we make today won't impact the herd for another 5 or 10 years from now. It is important that we start to focus on EnviroCow as part of the selection decision, even though the signals at the moment may be a little bit confusing.
Okay, thanks, Marco. I'm going to go straight on to Mike. Mike, a few questions for you. When analyzing a percentage infected milk TB by TB advantage trait, have you taken into account the age of the daughters or the granddaughters? Older bulls will have older offspring who will have had more opportunity to catch TB.
Well, the daughters, the longer the animals are in the herd, the greater the opportunity they have to obviously to get TB. But no, the statistics I showed were simply that the granddaughters of the best bulls, there were fewer of them having TB than the granddaughters of the worst bulls, and those bulls would have been contemporaneous. So, you know, those figures are comparable, yes, is the answer to that. But yes, clearly, I mean, you've got a situation, haven't you, where if a bull has got a good figures for survival, then his daughters survive longer and they had more opportunity to get TB. It's a sort of paradox, really. It's a paradoxical situation. But of course, what you want is actually animals not to get TB in the first place, because they have very short lives when they get TB.
Right, thank you. How soon do you think we might see the types of data be incorporated into AHDB genetic and genomic evaluations? What sort of time frame, Mike?
Yeah, I mean, we're currently in a field trial for both TB and for fertility. And bear in mind, those are existing traits that we already have. So the work that we're doing is looking at ways of improving the extraction of new phenotypes that we can then add into evaluations and make the evaluations more accurate. But going further afield using the same technologies, for example, we have people working at the moment looking at predicting feed intake and methane emissions using the milk spectra. So given that that would take you know, 1 or 2 years to do the research and develop the algorithms, you then see the data coming in 1 or 2 years after that. And that's one of the important things about genetic improvement. Marco already mentioned it. You know, the decisions you make today affect your herd for 5 to 10 years time. So A, you need to get it right, and B, if you're wanting to improve the genetic evaluations for the future, the sooner you start, the sooner you have these advantages, you know, down the line. So it seems like it's a long way away, but don't forget, the work on feed intake and gestation length was done years ago. That's produced the indexes that farmers are now using. So we have a very long time frame view of the world, which is why we try to do as much as we can as quickly as we can, because we recognize it takes a long time for it to get out and get used and have an impact.
Okay, thanks. Another one, Mike. Will farmers need to time milk recording to TB testing for best results?
Not particularly, but certainly anecdotally, when we've done some kind of field discussions, as it were, focus groups on TB, some of the farmers suggested that should the test that we're testing out, as it were, if it works, they would be willing to do more frequent milk recording to give them more opportunities to detect reactor cows. So at the moment, farmers generally milk record either every 4 weeks or every 8 weeks, mostly 4 weeks. But there's no reason why they shouldn't record weekly if they're specifically looking to detect animals with a particular disease or something.
Okay, thanks. Is there a risk that TB Advantage could be selecting for cows that are not displaying positive results rather than not being infected?
No, there's a published research. Whoever needs to know that, I'm happy to send you a peer research paper that demonstrates that is definitively not the case. We looked at breeding values calculated for animals that had lesions, as it were, in the abattoir, so they definitely had TB, and those where it was based on the skin test, and the distribution of breeding values overlapped perfectly. So there is very little chance. There's a theoretically small chance, but there is very little chance that that can occur. And even before it occurs, we have the tools to enable us to monitor it and determine what to do about it. So I think it's one of those things, you know, a little bit like inbreeding. You know, I'm not going to use those bulls because at some point in the future I might have an inbred herd. Well, it's a theoretical outcome, but if you manage it correctly, it won't occur.
Right. With each point of PLI now worth £1.58, will the industry be taking away the current pound sign or using the new figure for bull proofs?
You mean like a double pound sign or something, or some new currency for the UK post-Brexit? No, I mean, clearly having a pound sign in front was something that was put in, I can't remember, Marco, 20 years ago to actually, I think just when we went from PIN to PLI, they put a pound sign on and put it on the pound scale so that farmers could have a way of deducing why and how something was good. I don't see that changing, but that's clearly up to AHDB in terms of publishing the figures.
Just to add to that, I think that it's we've got to bear in mind that it is a trial, and I think we've really used it to hopefully show that PLI works, and I think it's a reassurance for ourselves that it works, and but to specifically say, well, it's worth £1 or 1.58. In general, the positive message, I guess, to take away from this is that genetics works. It may even work better than we predict it to be, but at the moment we're not thinking we're going to scale it up to 1.58 just to reflect the latest Promar figures.
Okay, thanks. And before I move on to Tim, just a question for Marco and Mike. Feed additives to reduce enteric methane emissions will soon start to be used. How additive do you expect the effects of feed additives and direct genetic selection for reduced enteric methane emissions to be?
Yeah, it's a great question, but we tend to have a stock answer to this type of question, which is genetics generally is additive, it's cumulative, and it's permanent. The alternative, for example, if you mix additives into cow's feed to reduce methane, you have to keep adding it all the time. As soon as you stop adding it, methane output goes up again. So, and that's a valid thing to do. That's a valid action. But of course, at the same time, we're over here trying to reduce genetically. So you can imagine that an animal that's been genetically improved for methane emissions, i.e. reduced methane emissions, when you add the stuff that you add into the feed, you should get even less methane emissions. So I would expect the two of those to be complementary.
Okay, thanks, Mike. Anything to add, Marco?
No, I think Mike made the point. I think it's, yeah, the main point that I think it's not one or the other. I think what we try and do is both, because I think the main one is really that permanent improvement, because if we're very good at breeding for it, in 20 years time, we could get away with not feeding the additives and save ourselves some money, because the cows are genetically good enough to not produce as much. So.
Yeah.
Okay, thanks. Tim, I'm moving on to you now. Tim, although other breeds and crossbreeds are small in numbers, would there be enough to show any patterns and trends if you did analyze them, or would they be statistically significant? Is the question.
Yeah, I think that the real strength of this analysis, and I think Marco and I have tried to see if it's been done elsewhere in the world, but we don't think it has. We think this is sort of a bit of a unique exercise that we've been able to do. But because we're looking at whole farm figures, it's absolutely vital to try and remove background noise by having a large number, because on a much smaller sample, other factors other than the genetics are going to have much more influence on the figures. So I think we'd probably just be up against lack of numbers in terms of trying to prove things more statistically, whereas with over 400 herds here and a very large cow population, we are able, I think, to infer conclusions which we simply wouldn't be able to do from a much smaller subset of data for other breeds, for example. I think Marco probably could comment on the degree to which you could infer from the data that we've actually done in terms of the other breeds, but I think just mathematically, I think doing it as such would be very difficult.
Yeah, I think that, well, to flip it, I think there's no reason to believe it doesn't work, because the way we do genetic evaluations, it's we don't, as I said right at the beginning, we're not biased by breed or country, so we're just assessing genetics. I think what this study shows is, does the genetics work? And okay, we've had to focus on Holsteins and PLI because that's where the data exists. But there's no reason to believe it shouldn't work when we look at other breeds. Certainly on the same system, we tried to look at the data to say, well, can we separate spring and autumn calving herds? But as Tim said, we just don't have enough herds to really give us the confidence to do it. We did do some work. Tim, just you may want to expand on it, but we looked at herds that have got a lower concentrate feeding versus higher concentrate feeding. So essentially trying to mimic high and low input herds to say, well, does genetics still work? And the clear message again out of that was that irrespective of the level of concentrate feeding, the better genetic herds are getting a better financial margin. And so that again gives us some reassurance that irrespective of the system, genetics does deliver financial. And I don't know, Tim, if you want to expand on that. But
yeah, basically we took the top and bottom 25% of the herds within the 400, which still meant we had over 100 herds in each analysis, and we looked at the ones with the lowest level of feed inputs and the highest, and effectively ran the same exercise for those subgroups rather than for the 400 plus herds as a whole. And yeah, the relationships held up really well, but within both groups. And that's something we had actually done in previous studies, and the same thing had been true. So I think that's probably as close an exercise as we can get as to whether some of the benefits that we've described within the overall study will translate through to lower input or higher input systems, and potentially across to other breeds as well.
Tim, could I just interject there and make a comment which was great? I was watching your slides, and I think it's important to note that you mentioned it, so I just want to reiterate it for the audience, that the higher merit herds had higher vet costs. But the important thing there was that their selection and their improved output gave them income that they could spend on preventative veterinary costs. And I think that was an important point, because a lot of people might misinterpret that if they don't pick it up, that therefore, you know, having high merit cows automatically means higher vet bills. Well, actually it doesn't. What it does, it gives you options that you can spend money on preventative medicine.
Yeah, and I think we've seen a general move within our figures as a whole towards preventative bills making up a high proportion of the vet costs. And yeah, I think I very much endorse that from the overall picture we're seeing as well.
Yeah.
Okay, thanks. And final one for Tim, and because of time, as the clock's ticking, what is your view on what this work means for low versus high input herds, and does it still hold true that genetics will provide for greater margin opportunities?
Yeah, I think probably that piece of analysis we've just referred to would be from the study. That's the closest where we can bring evidence to bear for that particular category of farms. So yeah, I think we can be very confident, having seen it within the study that we've done on that subgroup, as just described, that the benefits will be equally strong for low input herds as well as high input herds.
Okay, thanks. Well, I think I'd like to thank everybody for questions. There's been a lot of questions. We haven't been able to answer all of them, unfortunately, but we can get to you those that went unanswered. So thanks very much, everybody. I'm now going to hand back to Alex.
Thanks, Brian. So that leaves to me, I'd like to take the opportunity again to thank Marco, Mike and Tim for giving us their time and sharing their knowledge with us today. It's really great to hear about some of the huge range of genetic tools that we provide at AHDB to assist our levy payers with making their breeding decisions. We're really lucky to have a rich research and development pipeline to continue and to expand and refine these tools. And Tim has shown us that there's a clear economic benefit to using superior genetics at the animal and herd level. I'd like to echo Marco's comments earlier and encourage people to take a look at the dairy breeding information and tools that we have available on our website and to sign up for our herd genetic report if you're milk recording and you're not yet using it. As I mentioned earlier, our session recording will be made available to you within 24 hours, and we'd be extremely grateful if you could take a few minutes of your time to complete the feedback survey that will appear on your screen at the end of the session. Our final session of the conference takes place tomorrow at 11 AM and focuses on optimising young stock performance. So please join us again if you can, or if you're interested but too busy, then do register online to receive the recording after the event. With that, I think we'll wrap up. I'd just like to thank everyone who's attending for taking the time to join us this morning, and I hope you enjoy the rest of your day. Thank you very much.
Cheers.